Here is Dr. Shazia, Assistant Professor, Department of Zoology, Women University, Multan. Today's topic of this talk is the role of polyphenols in neuronal disorder. Polyphenols are more than 8,000 phenolic structures are currently known, and among them over 4,000 flavonoids have been identified. They exist with variable structures and are found in vegetables, fruits, grains, roots, tea and wine. As well as they are antioxidant, they have a very strong antioxidant effect, anti-inflammatory activity, anti-carcinogenic effect, and anti-aging effect that all studies carried out. Component of anthocyanin, which belong to the flavonoid family. Here you can see these are extracts of cyanidine. C3G extract that is mostly used in Korea, and as well as if we talk about the neuroprotective effects, so glutamate is one of the major excitatory neurotransmitter in the nervous system. So glutamate, when induced, increase the intracellular free calcium inside the mitochondria, and depolarization occurred. As a result, reactive oxygen species produced and lead to the apoptotic cascade and entered into the cell death. The neurotoxicity is widely thought to be a key event of neuronal damage. So if we focused on the key event of the neuronal damage, ranging from acute insult such as ischemia and traumatic injury to chronic neuroprotective disorders such as Alzheimer's disease and Parkinson's, so we can be controlled over the neurotoxicity. One of the major key role played by the zinc homeostasis in the neuronal death in vitro and in vivo studies carried out, the neuroprotective effect of zinc chelation is also play a major role. So zinc accumulation in degenerating neuronal soma was noted in forebrain ischemia. Rodent model were neuroprotective was observed with zinc chelation. So similar histological evidence was provided in rodent model of traumatic injury, which also demonstrated a neuroprotective with zinc chelation. So here is a zinc neurotoxicity. If we talk about their direct evidence for zinc neurotoxicity was provided by extracellular exposure of cortical culture to zinc, which lead to concentration and time dependent modes of apoptotic or necrotic neuronal death. So synaptic release of zinc is also thought to be involved in ischemic cell death. Here is to clarify how this flavonoid C3G factor glutamate induced zinc signaling in neurons. Here is glutamate induces intracellular zinc increases by a release of zinc from intracellular sources in cultured rat hippocampal neurons. In figure A is intracellular zinc induced by 100 micromole glutamate and then washed for 35 minutes. After washing, concentration of zinc and then 100 micromolar glutamate induced the intracellular zinc, and then wash for 32 minutes, and then use the 5 micromolar TPEN for 2 minutes. And chelator is calcium EDTA is used in same manner. Here is control in summary: TPN and calcium EDTA as a chelator of zinc of intracellular. And then we can see the final concentration of the zinc that released in response to glutamate-induced intracellular zinc from the here is different concentrations of cyanidine 3-glucoside from 100 nanogram per ml to 1 milligram per ml used, and intracellular calcium concentration dependent inhibition of glutamate induced intracellular zinc increases that was inhibited by using different concentration. The final concentration IC50 value was determined by normalized response. IC50 value was 14.1 microgram per ml, and this concentration was used in further experiments. Here is about this effect of cyanidine 3-glucoside, intracellular calcium chelator BAPTA-AM and intracellular zinc chelator TPEN on glutamate-induced intracellular zinc and intracellular calcium increase was here. We can see in first in control A 100 micromole glutamate was induced, the intracellular calcium and intracellular zinc, and here is zinc was used by inhibition of cyanidine 3-glucoside in A2. It's a very significant effect of cyanidine 3-glucoside. And then on zinc there is also effect of this in presence of chelator, and then there is chelator of the zinc was used. So in calcium is in same manner, intracellular calcium was induced by glutamate 100 micromole and 100 micromole glutamate, and then effect of C3G was here we can see it ameliorates the effect of induced or inducer, that is glutamate, and then in presence of chelator of BAPTA and intracellular calcium and zinc. Here is summary. There is a significance of cyanidine 3-glucoside on the intracellular calcium chelator and intracellular zinc chelator in response to glutamate induced zinc intracellular zinc. So effect of cyanidine 3-glucoside as an antioxidant Trolox and the reductant DTT on glutamate-induced zinc intracellular calcium and zinc. In response to here is control of A1 is glutamate, and then there is glutamate and cyanidine 3-glucoside in presence of antioxidant Trolox and DTT, and there is a significant effect in both cases in the zinc and calcium. So glutamate receptor mediated intracellular calcium increase has been reported to induce formation of RBS, and glutamate induced zinc increase is caused in part by intracellular calcium induced formation of RBS. Here is effect of cyanidine 3-glucoside, Trolox, DTT, BAPTA-AM, TPEN on glutamate-induced formation of RBS. And here is control and the cyanidine 3-glucoside, Trolox, DTT, BAPTA-AM, TPEN on here that is with parallel to the vehicle. Inhibitory effect of cyanidin 3-glucoside, BAPTA-TPEN, Trolox and DTT on mitochondrial membrane potential (MPP), that is the mitochondrial membrane potential, in glutamate induced for 35 minutes, and there is FCC effect, and here is BAPTA-AM and Trolox effect was checked. In both cases, in rhodamine 1, 2, 3, and then effect on C3G and FCCP as well as TPN. Effect of cyanidin 3-glucoside, Trolox, DTT and TPEN on glutamate-induced neurotoxicity in culture pure rat hippocampal neurons. Cell survival we can see here is in TPN in presence of TPN and DT and Trolox as well as cyanidin 3-glucoside. You can see these are antioxidant effect, which here is a significant effect of cyanidin 3-glucoside on the cell survival. In figure B is where we got in 10 microgram glutamate that induced the intracellular zinc and calcium, and cyanidin 3-glucoside 15 microgram, as well as here is induced glutamate plus cyanidin. Effect of cyanidin 3-glucoside on proliferation and cell death through calcium homeostasis of endoplasmic reticulum in human prostate. So these polyphenols not only affect on the neuronal cells, but as well as they also affect on the significant effect on proliferation and cell death in LNCaP prostate cancer cells. From 24 hours to 6 days exposed this, and LDH release was assessed and number of cells. Its proliferation and cell death in LNCaP in the presence of epidermal growth factor that induce the cell proliferation. First I have to elaborate. In presence of different concentrations of cyanidin 3-glucoside from 1 microgram per ml to 100 microgram per ml of C3G, the LDH release and number of cells, as well as the effect of cyanidin 3-glucoside on basal level of ER calcium modulation on LNCaP cells. Here is the endoplasmic reticulum calcium in 34 nanometer to 380 nanometer is in the presence of epidermal growth factor and hepsigargen that release the calcium in the homeostasis inside the cell, as well as in presence of ER calcium and without epidermal growth factor. So here we can see the LNCaP cells shows their response very significantly, effect of cyanidin 3-glucoside. So in summary, its reproducible glutamate-induced zinc increases was elicited by glutamate. Cyanidin 3-glucoside also affect on in response to reactive oxygen species. As well as the cyanidin 3-glucoside inhibit glutamate-induced intracellular calcium increase through a release of zinc from intracellular sources in cultured rat hippocampal neurons by inhibiting calcium-induced mitochondrial depolarization in the neuronal cells as well as in prostate cancer cells, in formation of ROS, which is involved in neuroprotection against glutamate-induced cell death. So polyphenols significantly affect on the neuronal cells and prostate cancer cells. Thank you so much for listening this presentation. If you have any